Hello students, this is a plant cell. Although I have given you the introduction of the chapter, the cell, but here I would like you to show a model of a plant cell where we can see the different things properly. You see here, this outer one, this is the cell wall the student has tried to make it, and this inner one is the cell membrane, because they have the two-layered wall in case of plant cell. Now this one which the student has made with the help of green color, this is the chloroplast which is being indicated. This one of course is a mitochondria. This one is a granular cytoplasm. So they are sometimes known as your cell inclusions. Now this one represents a endoplasmic reticulum, which is a smooth endoplasmic reticulum. As you can see on the tubule of the endoplasmic reticulum, there is no dotted structure. There is no ribosomes present over there. So this is a smooth endoplasmic reticulum. Now let us come to this one. Here you can see various dotted structures. I have tried to magnify it as much as possible. So this one is called as your rough endoplasmic reticulum. Now a plant cell has a large vacuole. So you can see that how large the vacuole is, and because of this almost central vacuole, large vacuole, because it's an organ of storage for plants. So in them the nucleus is towards the periphery. So this is the nucleus. The nucleus is halved, and inside the nucleus you can see one circular object is there, which is called the nucleolus, which is associated with the synthesis of the RNA as well as that of the ribosomes as well. Now this one is, which is near to that of the nucleus, is indicated as Golgi body, but in case of plant cell we will not be calling them as Golgi bodies, because they don't have a definite structure as it has been made here. They are lamellae, but they are not in such a manner that they have a cis phase and a trans phase in case of Golgi bodies in case of plant cells. So they are called as your dictyosomes in case of plant cells. And of course this one is a mitochondria, which I have shown you before only. So this is a generalized view of the plant cell which we have seen it today, and now I will be showing you the animal cell as well. Now this is an animal cell. The difference you can see it very clearly. In this model of animal cell, you can see that the cell is only bounded by a cell membrane. It doesn't have a cell wall. Okay, so it is having only the cell membrane, and inside you can see that the cell membrane in some places have some invaginations or finger-like projections, as we see in case of certain cells in our parts of the body, like that of the intestine and all, where they form this finger-like projections, microvilli and all, for the absorption of the substances. Okay, now next, if you see again very closely, now this one is a Golgi body. Now here you can see that the Golgi body has a specific structure having a cis phase and a trans phase. Okay, so from where that is the ribosomes or other materials from the nucleus comes out, and they undergo the processing through this lamella and are pinched off from the other surface of the Golgi bodies, and can be used for the membrane biogenesis, or they can be used for any other kind of cellular work and all. Now this represents the mitochondria. Animal cell has a lot of mitochondria because they need a constant supply of energy by means of the cellular respiration. Now this is a vacuole. In case of animal cell, the vacuole is very small. Either it is absent or it is very small. And this is of course a central nucleus, unlike the plant cell, in which we have found that the nucleus was peripheral. The nucleus is bounded by a nuclear membrane. Here the membrane is not shown so properly, having some nuclear pores in it. But yes, you can just identify that it is a double membrane structure, and this round surface indicates the nucleolus. Okay, they represent the nucleolus, and inside the nucleoplasm, and these are the chromatin threads which are being depicted over here. And surrounding the nucleus, you can find some structures which are tubular structures having some dotted structures over them, and they are called as your endoplasmic reticulum. Okay, and here, see, just above the nucleus, you can see one cross-like structure is there. Now this represents the centriole. Okay, this is centrosome containing the centrioles. There are two centrioles which during the time of cell division will take part in the formation of spindle fiber, through which the chromatids will be held together. And of course there are some microfilaments within the cytoplasm, so they are the inclusion bodies which are found in case of the animal cell. So this is how the plant cell and that of the animal cell is different from each other, as well as having some common characteristic among each other. I hope you understood the difference between the plant cell and the animal cell well.